Thank you. So today's webinar will hopefully give you an overview of what we're working on for metrology for drug delivery. I'll just mention my experience and background. Originally, I did my PhD at Edinburgh University, and that was on microflows. I've then worked at NEL for about 14 years. I kind of scaled up and was working oil and gas at much larger flow rates and larger size pipes. And then I'm starting to apply my knowledge from the PhD for metrology for drug delivery and working in the microfluidics area again. So this webinar, I can't cover everything on the project because it is quite a large project, but hopefully it will give you a general overview. And if you need more information, or you're interested in the project, then you're very welcome to contact me. So in terms of the content, I'll give a brief background to the project. I'll then cover some of the calibration techniques and the development of these, then moving on to testing actual drug delivery devices, before moving on to more complex setups of multi-infusion drug delivery systems, and then talk a little bit about the dissemination of the information from the project and impact. So if we look at first, why are we doing drug metrology? There's actually a very high percentage of patients when they go to hospital actually receive IV therapy, and this can be to deliver medication, fluids, nutrients. But there's also been numerous injuries, adverse health effects, and even deaths from issues with using infusion pumps. So it does highlight there's significant safety issues, and generally this isn't talked about, but it is known within hospitals. So if you are going to go to hospital or you're using any of these devices, you want to have confidence that the delivery of the medication is precisely controlled over time and that the delivered dose is accurately known. And this is especially important if you have high concentrations of critical drugs, and they are quite potent. You want to make sure that you do have regular calibration and maintenance of all your infusion equipment, so you can quickly identify then if you have any issues with your equipment, and this also minimizes any health and safety risks to your patients and ensures that they get the correct dose. So you can see in the picture, this is a setup of several syringe pumps and an incubator. We'll discuss the multi-infusion setups at the end of the presentation. So this is quite a good example of issues that can come about from infusion pumps. It's quite well documented in the news. It was an issue with insulin pumps by this company, in which they had to withdraw over 300,000 insulin pumps after there was reports of injuries and at least one death. And it has caused a lot of issues for the company, including like lawsuits, issues for the users, including diabetic comas, other injuries. So it is quite a high profile issue, and it's been well documented, but other issues with infusion pumps haven't been so well documented, but it just kind of highlights why you want good metrology. So what are we doing? NEL is part of an EMPIR research consortium which aims to characterize and improve dosing accuracy of existing drug delivery devices and multi-infusion systems. And this is to enable traceability of different measurements like volume, flow rate, pressure, inline sensing capability for very low infusion rates. And what we're looking to do is by developing new calibration methods, which I will describe, and also by expanding the existing metrology infrastructure. There is a lot more information on the project at this website, and I'll also give you the website at the end of the presentation as well. So in terms of the consortium, there's a large number of partners which are involved in this research based across Europe. So we have a wide range of skills from each of these partners, which is actually supporting us. So it's a range of universities, national measurement institutes, and also instrumentation providers. I'll give a general overview. Additionally, the project will also investigate the effects of fast transient flows on dosing response and also the physical properties of different liquid mixtures in infusion and also occlusion phenomena in multi-infusion systems. What I mean by that is if you get a blockage within the delivery of the drugs, then the pressure can build up and cause issues. You want to be able to identify that there is a blockage and the drugs or medication isn't reaching the patient. Also covering the development of a new micro pump. I've already sort of covered that, this is very important for drug metrology, but it's also important that these applications and techniques that we've developed can also be used for microfluidic applications and other applications, for example, organ on a chip, as well as drug delivery. So when we talk about calibration of infusion devices, what I have at the very top of the screen, this is an example of what an syringe pump looks like. So you've got your plastic syringe and then you can then program in the actual flow rates that the syringe should deliver. The graph shows if you start up the flow rate, it'll take a while and there will be a delayed startup time until you achieve your actual flow rate. So this is what we're looking at when we're doing calibrations, how quickly it gets to the target flow rate and how stable it is. So typical infusion pumps which are used, you have your syringe pumps, peristaltic, you can have gravity controlled insulin, pen pumps. There's a huge number. What we have focused on is critical applications which require a high degree of accuracy, and these may be used for drugs with very narrow therapeutic margin, drugs with a short half-life, and any high concentration drugs or drugs which are given to very, very small children. But the calibration principles, they can be applied to any type of pump. We'll move on to the development of the calibration techniques. So we have been looking at the development of infrastructure for ultra-low flow rates. And what I mean by ultra-low flow rates is techniques which can measure down to 5 nanoliters per minute for steady and fast-changing flow rates. And I'll give a description in the next slide of how you can visualize that very, very small flow rate. We're also wanting to establish robust and realistic uncertainty budgets. So our targets for this are 1% for steady flows and 2% for fast changing flows. These are actually very ambitious targets considering the flow rates we're going down to. And by the end of this year, we hope that we will be able to validate what the uncertainties are. And we are going to be, as I said, validating these. We're going to do an inter-comparison exercise. So it will compare all the different facilities and techniques which have been developed by the partners. So for ultra-low flow rates, we are looking at flow rates from about 100 nanoliters per minute down to 5 nanoliters per minute. And this is tiny, tiny amounts. So if you imagine a drop of liquid and say that the diameter of that drop is about 2 millimeters, to then achieve a flow rate of 100 nanoliters per minute, the time to develop that drop size would take 50 minutes. And if you're down at 5 nanoliters per minute, the time to develop that drop would actually take you over 16 hours. So it gives you an idea of how difficult it is to try and develop these techniques and get very, very low uncertainties for these ultra low flow rates. These are some examples of like nano-flow applications that will go down to nano flow rates. So the first one is implantable infusion pumps, and these can then be used to deliver medication, different drugs to very specific targets in the body. And because they're implantable and they'll deliver the medication to the target, it means you can use much smaller doses of the drugs. So these can go down to 33 nanoliters per minute in this example. Another example is the use of insulin pumps, and these can go down to very, very low flow rates in the nanoliter per minute range as well. These are becoming a lot more common to be used, and as the original example I showed you, there was an actual issue with insulin pumps which did cause significant safety issues for the users. So you want to make sure you have confidence in using these devices. There's been a lot of research done for steady flows, but we're actually looking at more challenging, dynamic, fast-changing flow rates within the project. And what I mean by that is that the flow rate can change over a period of less than a second. And what we want is our facilities or our calibration techniques to be able to track and monitor if there is fast changing flow rates. And this is because we want the techniques to be able to identify and track if there's any spikes in the dosage of drugs. Most of the manufacturers do claim that you'll have a steady concentration and delivery of drugs, so the flow rate should not fluctuate, it should stay steady, but we need techniques to be able to identify if there is going to be an issue and if you are getting spikes or changes in the dose. So we will be monitoring the techniques and looking at the response time and how quickly these techniques can monitor that, so we can robustly test the different pumps and infusion devices which are on the market. So traditionally, the gravimetric method has been used, particularly for higher flow rates, or like what we would call maybe microflow rates. So it involves, you would have your flow generator for drug delivery. This could be like an insulin pump, syringe pump, pen pump. We may have a device under test as well, or instead, this could be our infusion device analyzer. I'll explain a little bit more about that later. Or it could be a flow meter, and sometimes you could have a flow meter in your infusion line to be able to monitor. So there are limitations with using the gravimetric method, in that it's very difficult to go down and use it for nano flow rates. It's been well established at much higher flow rates. This is an example of a gravimetric calibration setup that we have at NEL. So we have our syringe pump, we have an inline flow meter, we have different pressure sensors, temperature sensors, we've got our precision balance, and we have our data logging instrumentation. So this is actually for much higher flow rates, from 1 microliter to about 10 milliliters per minute. But when we go down to much lower flow rates, in the nanoliter per minute, we have to use a different setup where we're using a much smaller balance. So this is a setup that has been done at one of the partners, DTI in Denmark. They are using either a 6 gram or a 10 gram balance. They actually have it in a chamber. It's more difficult to see it in this picture, where they can control the temperature, humidity, make sure there's no drafts, which would actually affect any of the measurements. They have little flow sensors in line, different instruments that will be able to record and control the flow rates. So they've been able to establish this to measure steady flow down to 15 nanoliters per minute and dynamic flow down to 20 nanoliters per minute. Another technique that we have been investigating is using optical measurement standards. One of them is using a pendant drop and the formation of the pendant drop. So for example, if you have a glass capillary and you have liquid flowing out the end of that to form a droplet, what we're looking at is the rate of change of the volume of the droplet over time. And we can relate that back to the actual flow rate. So you need a high resolution camera, you need special lighting, different optics to be able to image it. Generally, the more expensive your camera, the better imaging you're going to get. So in the bottom, you can see pictures of the droplets and different optical techniques that we're actually using to be able to image that better. So this is under investigation at CETIAT. You can see that in the top picture, and also at IPQ. You can see that in the bottom picture. Another technique is interface tracking. This has been set up. The images I'm showing here are from CETIAT in France. What it is you can have a glass capillary and actually image the the movement of the interface between the water and the air, and then calculate the velocity change and relate that back to the flow rate. So in the image on the left, we can see we've got various setups. We've got a syringe pump, we've got high resolution camera, different optics. We also have a translation stage, so we can move the equipment to get better imaging and to actually track the movement of the interface. So on the right hand side, you can actually see in more detail from the diagram the actual setup of this. And these images show the actual interface, and using the camera we can then track that as it moves. We have information on the time over which the images are recorded and the displacement, so we can work out the velocity and then calculate the flow rate. This system's also been set up at Lübeck and at IPQ as well. So we can see on the left-hand side the tracking system that's been set up at Lübeck, and on the right-hand side the tracking system that's been set up at IPQ. So there's different instrumentation which is being used, and it's good that it's actually been tested with different partners to verify how well it works and to be able to compare the measurements. Another technique which could be used is almost like a piston improver, and this is used at much higher flow rates. It's been set up at METAS, RISE and IPQ, but using completely different equipment. So at RISE, they're using a commercial syringe pump, which you can actually get information from that as the plunger is displaced. METAS have built their own custom-built syringe pump, where they can record and get information on the displacement and relate that back to velocity movement of the syringe. And IPQ are using a more advanced technique using interferometer, which is directly used with the actual syringe pump. It is a more expensive method, but it does give you much lower uncertainties. Another technique is microparticle imaging velocimetry, or the short name micro-PIV. So I think you should be able to see the video on the right hand side. So how this works is we've got small particles which are suspended in the flow, and these should be traveling at the same velocity as the surrounding fluids. So if we can actually track these particles, it gives us information on the velocity of the fluid, and we can relate that back to the flow rate. So this is the setup that's at Strathclyde University in Glasgow. So what they have in the image on the left-hand side, you have your microscope. They're using a pressure control system. This is to accurately control the flow rate and eliminate any fluctuations, and using a high-speed camera. It's quite difficult to see the little microchip which they've designed and fabricated in-house. So the image at the center shows this little serpentine microchip, and the diameter of these channels is 50 micrometers up to 100 micrometers. So it's very, very, very small. And the serpentine design allows them to move between each of the channels to be able to then monitor the actual velocity. So if we look at, so the image I'm showing now just shows the particles in the flow and being able to track that around the serpentine design. We're now seeing some results, and what we can see is it is actually working down very well down at 5 nanoliters per minute, and it does seem to be very quickly tracking any changes in the flow rate as well. And because we're using a pressure control system, you can quickly change the control of the flow. The PIV method's also being developed at Hans Schickert, another partner in Germany. They're using a holographic PIV system. You can see it's a much smaller or more compact setup. They've got an LED light source, pinhole. They've got a small carrier, and they've got an imaging sensing capability within this setup. You can also see the little flow chip that they're using as well for this. So I have briefly covered the Nano-flow developments. We have got a comprehensive report on all these techniques for the steady and the dynamic flow rates, and that's free to download. I've given the information here. So it's a 78-page report, and it covers all the information, provides pictures, descriptions of the techniques, how you do the uncertainty calculation. So it can be very useful for people. And also we did have an online workshop in November last year, and also these presentations are available, and it'll talk you through all the techniques in more detail if people are interested in that. So the next stage is we have just begun doing an intercomparison exercise, and this is where we have two thermal flow meters and a syringe pump, which is being circulated around all the laboratories in Europe, so we can establish and compare the results from each facility. Because what you want to ensure, if you test a drug delivery device in one organisation, you're going to get the same results and values as if you test it in another organisation, and there's no bias in the results. So we're going to move on to testing the drug delivery devices. So this is another work package where we have identified the needs for the metrology infrastructure. So we sent out a survey and questionnaire. This was sent out to different medical departments, commercial companies, hospitals, just to make sure that the testing that we are developing and what we're doing is actually fit for purpose and actually needed. What we're wanting to do is develop and validate the calibration procedures and generate guidelines that other people can apply these techniques. We've also been looking at the development and characterization of a novel microchip pump, and you can see the diagrams in the bottom just showing the protocol that we're going to use for this. If we look at the testing procedures, we've selected 4 devices, and this was in discussion with the people that helped with the questionnaire on what would be most appropriate to use. So we've selected a syringe pump. This is widely used in hospitals to deliver very small amounts of fluids to patients, and it should have a very high accuracy. There is calibration procedures which are described in this standard. These are mainly being used by manufacturers, but there isn't any calibration procedures for when it's been in use. This is not documented anywhere. As well as using water for the calibration procedures, we have been advised to try to use some actual drugs. So some of the partners will be using dopamine, saline solutions, other drugs, just to make sure that the measurements that we do are fit for purpose, rather than most calibrations have always been using water. Another device that we're using is an infusion device analyzer, and this is used to verify the accuracy of drug delivery devices. They're normally calibrated by the manufacturer before they are sold, and then subsequently calibrations may not be performed, and there's no available documents covering the calibration for users. If you look at the images, if you look at the images at the very bottom, so you can see in the center there is a syringe pump which is set up. The liquid, the water is flowing from the syringe pump through the device analyzer and into a beaker. So the infusion device analyzer is being used to test and calibrate the syringe pump, and that's how it would be used in testing laboratories or hospital labs to verify the other equipment. But you want to make sure that your infusion device analyzer has actually been calibrated and has a low uncertainty before you're using it to calibrate other instruments. Another device that we're going to test is a pain pump, and this is an implantable constant flow pump which is used to give medication directly to the spinal cord, and hence the conditions can then be controlled with a much smaller dose than what you would need if you were taking medication orally. Again, devices are checked and calibrated by manufacturers, but there's no subsequent verification, and there's no documents available that explain how you would verify the performance and do calibration checks. And you can see that we're going down to very, very low flow rates for this device. The next device we're going to be testing is an insulin pump. It's widely used by diabetic people. Again, they are calibrated and checked by the manufacturers, but there may not be any subsequent verification, and there's no documents available on how you can do the performance checks. With most of these devices, you expect the dosing to be continuous. So what we want to do is have techniques that we can check that we are getting a continuous dose and we're not getting any spikes in the dose. With the insulin pump, we'll be testing that with water, but we're also wanting to test it with an insulin substitute, because it's always best to be able to test devices with the fluid that they're going to be used with. You can see that we're going down to very, very low flow rates, down to 4 nanoliters per minute for these devices. So as I'd said, there's not much information available on guidelines on how you do the calibration of drug delivery devices. So once we test these devices, and these devices are getting sent round the European laboratories, and so we can actually compare the measurements and compare the techniques that we're using. We'll then develop guidelines which should be freely available on our website and other platforms, and these will then feed into standards organisations for dissemination. If we move on to multi-infusion drug delivery systems. So we've covered quite a lot about calibration techniques for single pumps, but things actually get a lot more complicated when you're using multiple pumps at the same time, where the fluids are then commingled or combined before a single injection point into the patient. And these setups are very common in hospitals, where you have different drugs that need to be administered to a patient at very different flow rates. So we're going to cover some of the issues and complications with using multi-infusion setups and the impact on the flow rates. So this is just an example of what maybe a typical multi-infusion setup would be like. This is quite a simple setup. So we have 3 syringe pumps and syringes. And then the fluid is flowing from that. We've got here some check valves. This makes sure that there's no backflow of the fluid back into the syringes. We've got a mixing manifold where the fluids are commingled before flowing into a single infusion line. You may have filters. This is to remove if there's any air bubbles or any other issues, and then into a catheter and then into the patient. In reality, it could look something like this. So it can look quite messy. So we've got an image which shows the different syringe pump, different infusion lines. You can see different filters. You can see the mixing manifold. So it's got different colors for each of the inlets, and these are then mixed and then into a single infusion line before going into the patient. So some of the issues that we can get with multi-infusion setups and what affects the dosing accuracy, you can actually get different interactions between the pumps. So if you change the flow rate on one pump, it can actually affect the drug delivery from another pump. You can get what we call the dead volume effect, and this causes push-out effects. So at the image at the very top of the screen, what we have is we've got a blue, clear and yellow drugs, which are then coming from a syringe and are then being mixed into a single line before flowing into the patient. So the dead volume is actually the volume in the commingled or mixed infusion line before it reaches the patient. And I'll come on to talk about that and have some animations to describe why this can be an issue and what the push out effects are. We can also get issues with compliance, and if you look at the plastic syringe in the center of the presentation, what can happen is if you increase the flow rate, what can happen is, because we're dealing with plastic components and the elasticity of these can actually expand. So they'll absorb the increase in the volume from changing the flow rate. So if that's absorbing that, it's not pushing out the drug at a higher flow rate instantly. There can be a delayed reaction. You can also get resistance from very narrow tubes if you've got catheters, other devices. That can cause resistance issues. You can also get air bubbles, you can get viscosity effects, and you can get backflow. What I've got on the bottom right hand side is these are some little manifolds for mixing different infusions. So if you have, these are for a triple infusion and for a double, and you can see what the dead volume would be. So all these issues can cause a delay in the correct drug administration, and it can also cause temporary overdosing or underdosing. If we look now at the compliance effect, so what we see here is we've simplified this down. So we've got our red drug and our blue drug, and these are both flowing out at 1 milliliter per hour. So at the same flow rate, they're going into the mixed infusion line and traveling at the same velocity. Then if we have one of the drugs is actually increased or doubled the flow rate, what happens first of all is we have a compliance effect where the syringe and other components can expand and absorb that increase in the flow rate and the increase in the volume that should be pushed out. So we can see in the graph at the top, there's not an instantaneous increase in the flow rate. What it is it's quite gradual and it builds up the flow rate because of this compliance effect. However, this can also then have an effect on the other syringe in that the increase in the pressure from increasing the flow rate in the red syringe will actually cause an expansion and compliance effect in the blue syringe. And because that's absorbing and expanding, what we're getting is a decrease in that flow rate, which is being delivered to the patient. So overall, the effect from doubling the flow rate in the red drug is that we get a gradual increase in that flow rate delivered to the patient. But with the blue drug, we see that we get a reduction initially before it goes back to the correct dose to the patient. So that describes the compliance effect. If we then look at the dead volume effect, so we see again we have our blue drug and our red drug, and these are flowing at the same flow rate, so it's 1 milliliter per hour. So in the infusion line into the patient, we see we get 50% blue and 50% red, which is being delivered to the patient, and this is at flow rate of 2 milliliters per hour. So if we then increase the flow rate in the red, which we'll double that to 2 milliliters per minute, what happens is after we have the compliance issues and other things happening is when we go into the infusion line, which is going to the patient, we can see that initially it was 50% red, 50% blue. We've then changed the flow rate, doubled that. So then it's now 67% red, 33% of the blue. But because of the dead volume effect, it still has to push out this 50% of the red and the blue to the patient. So if you have a very long infusion line to the patient, it's going to take a considerable period of time before the increase in the red actually reaches the patient. So this is what we call the dead volume effect or push out effect, is that time delay, and it's related to the volume in that input in the infusion line. So it can be very difficult to administer the correct amount of drug, and you have to be careful if you're using very fast acting drugs or very potent drugs or drugs that have a very small therapeutic range, in that you need to keep it within very, very tight tolerances. So this can cause complications when you have multiple syringe pumps which have different flow rates, and if you need to change the flow rate or a doctor decides to double a medication, then it may take some time before that reaches the patient and you get the therapeutic effect. So within the project, we have been working on the design and characterization of multi-infusion systems. So we have done surveys, had interviews with clinicians, hospital staff on what is the best practice, what's their recommendations, and also what problems have been identified. So we've done this survey of the best practice. This includes issues we've already covered, but also like pressure alarms. So if you get a blockage, an alarm will go off, which will highlight to medical staff that there is an issue. Air bubbles, viscosity effects. What we're looking at is, well, what are the effects of viscosities on different mixing and flow, and also the delivery of the drug concentration over time. We're also looking at pressure equalizing devices and check valves. So there was a model developed in the previous project, and all this information is again available on the website. So we're looking to extend that multi-infusion model to include other issues, for example, check valves, different concentrations, mixing, to then investigate the effect. We are going to be setting up a clinically representative setup for multi-infusion system, and this has come from our interviews with the hospital staff. So we've developed a test matrix and we have all the equipment, and it's the equipment that they would actually use in a hospital that we are going to be trying out and testing, and the actual syringe pumps. So we're going to characterize 3 different medication schemes that actually represent real patient cases. So the image here just shows how complicated multi-infusion systems can be. You can have multiple pumps delivering drugs to a patient. So you want to make sure that the drug is getting delivered and it's accurately being dosed to the patient. We're also going to be using computational fluid dynamics modeling to investigate the mixing and other effects. This is a setup at Lübeck University, one of the partners. They've got some medical syringe pumps you can see in the back where they've got blue solution, red solution, clear solution, and these are the infusion lines that you would actually use in a hospital, and a mixing manifold that you would actually use. And they're actually testing if we change the flow rate of one of the syringes, what the actual impact is on the concentration. We've got a spectrometer set up here at the bottom of the picture to be able to monitor the changes. So that's given an overview on some of the technical aspects of the project. I'm going to move on to, well, how are we disseminating all the information and all the data that we collect? We are going to be developing new calibration procedures and guidelines on the drug delivery systems that we test, the calibration techniques that we develop, and the research that we're doing on the multi-infusion systems. So these will be available on the website and other platforms. We are then going to be providing this information to the standards organisations and bodies so it can be taken up and implemented in reviews of the standards. And these are just some of the examples of standards that we are going to be working with some of the working groups on. I'm going to come on to an example of the issues with using infusion systems in hospitals, and this is looking at specifically the impact of COVID-19 because it's quite relevant to this year and has affected everyone. So some of the challenges that COVID-19 brought to hospitals, especially for the use of infusion pumps, was the challenges included the reduction of exposure of healthcare staff to COVID-19. There was also issues with the preservation of personal protective equipment. There was shortages of other equipment, and also there was issues around waste management of quite scarce medications that are needed for COVID-19 care and other critical drugs, and also even like the management of staff shortages, reorientation of staff to areas outside their usual area of expertise. So some of the measures that had to be taken by some hospitals were actually to install and use the drug delivery systems outside of the patient's room. And this then reduces the exposure of healthcare staff to COVID-19 and other issues. If there wasn't sufficient equipment or there might have been issues with using drug delivery devices that were associated with lower accuracy, and this would have different clinical risks. If there wasn't sufficient syringe pumps available, were other pumps being used instead? There was also issues with having to postpone the deadlines for maintenance and calibration equipment, even equipment that is used for critical use. So we did identify quite a few of these issues, and we produced a case study. We've got a report, a flyer, presentation, which outlines some of these issues and gives guidance on how you can mitigate that. So that's freely available on our website. This is an image showing the impact of the COVID-19 on some hospital wards. In this case, they actually have the infusion pumps and a lot of the medical equipment outside the patient's room, and this is to reduce the exposure of the staff. But what I was describing with sort of things like a dead volume effect, so the infusion line going to the patient is going to be much, much longer, so you're going to get more of an impact from that. So if you're changing the medication or increasing, decreasing that, it's going to take a much longer time for that drug change to reach the patient and get a therapeutic response. So it gives a quite nice overview, and if you're interested, that's available on the website. We've also produced a guidance note on the calibration of medical infusion pumps. This is available freely at this website. So it provides an overview of performing a calibration using a gravimetric technique, but it also refers you to the correct standards to use as well. It's mainly been focused on syringe pumps, but you can also use it for any other calibration of other devices as well. And it's using the gravimetric technique. So that covers what we've been doing on the project. It is quite a brief overview of everything. It is quite a large project with a lot of partners, so we can't cover everything. But hopefully it's been insightful for you, and if you would like any other information, then everything's freely available on the Drug Metrology website, which is shown here. Otherwise, you're very welcome to email me for more information. On the actual website, there is like a news list, so any new publications or anything is actually available on that and highlighted on that. And I'd just like to say thank you very much for your time and listening to the webinar. Thank you.